So we were asked to switch talks, that's why I'm the next person. And then we'll have the Taxator talk afterwards. Right, I've been here last week already, so I'm not introducing myself largely. I will tell you a bit more about, so last week I said a few words about CAMI. And now I'm going to tell you a bit more about the research into biological questions that we do. And and how we use computational methods to try to gain some insights into the microbes that we sample with metagenome data sets. Right, so I'm going to start at a point of my presentation where I stopped last week because I want to save some time. So I told you last week about basically that you do shotgun and 16S analysis for taxonomic profiling. And where I stopped at was taxonomic binning. So I'm going to tell you quickly about that. One of the things that we do in my lab, I'm doing this because I just have 45 minutes, 30 minutes instead of 45. So one of the things that we did early on was develop a tool for the problem that Alex also just described, which is this problem of finding contigs or sequences that come from the same taxon. And in one of the early days, you just had single samples. And so the information that you could use for this problem in the absence of closely related reference genomes was k-mers, so short oligomer profiles, because these are more similar for fragments from the same genome than for fragments across genomes. And using basically, if you have some sequences of a known taxon that is in your sample, you can use this little data to train a machine learning method that is able to accurately classify even if there's just little data. And that's true for support vector machines. And so we use SVMs to build multi-class classifiers for modeling different taxa, and included that at different taxonomic ranks. And for instance, then we're able to retrieve data from abundant taxa in a wastewater treatment plant, enhanced biological phosphorus removing sludge. That was what this community is capable of. And so the size of these circles here shows you how much data proportionally we got out for different taxa in the sample using this composition-based approach. So this was a method developed with Phil Hugenhaus and Isidor Rigutsos while I was at IBM. And it's still in corporate, it wasn't released, the codes. So we re-implemented a framework. When I moved to Germany later on, we implemented a tool which we called PhyloPythia-S. And this uses a bit more fancy support vector machine, which is called a structural support vector machine. So with that, you can directly model a tree, basically, instead of just individual classes. And you can model in the tree, so every node in your tree is an output class, and what this model can take care of, that different nodes are related. So if you look from the root, they share parts of the path, and with this framework, you can basically model these correlations with a machine learning and build a classifier. And at the moment, so all these, since then, one thing we also focused on was making the tools faster because the data sets kept increasing in size. And the latest thing what we're working on right now is what we call PhyloPythia-S plus. So the S is for the structural, and the plus is because now something that we had to do before manually is done automatically, namely if for a given metagenome sample, finding out which taxa should be included in the model. So in a multi-class classification framework, you cannot include every possible taxon on Earth, but based on 16S, an expert usually says, these are the most relevant taxa I want included in my model, and this is some training data I give to you for them based on marker genes, for instance. And here this tool automatically screens the sample for marker genes, for all kinds of marker genes, using models defined originally by Amphora, so from Jonathan Eisen's lab, and then defines based on that what to model. And since then, and Johannes, what he's going to talk about is a tool for that doesn't use the k-mer approach, but sequence similarities for taxonomic assignments. So we just wanted to have something complementary that makes use of an alternative source of information. It has different pros and cons. I'm not going to go into detail at the moment. It's better for unassembled data. So if you have short reads in complex environments, you might want to use tools that use sequence similarities. So PhyloPythia in comparison to PhyloPythia-S, just an overview of the framework. we had here the multi-class models for every different taxonomic rank. And because we have a tree of life that we want to assign to, basically, if we have one particular sequence fragment, we have it classified by the multi-class models at all ranks. And we have at each class node, at each rank, a class unknown, which is trained with all the data that for classes that we don't model explicitly. And then we assign it to the taxon, so to the model at the lowest rank, what wasn't assigned to unknown. And usually the assignments of the different classifier agree. And this gives some flexibility. You can just plug in different class models down here. And now in the new framework, we model directly the taxonomic tree. So here the output is a path in the taxonomy, but also it could be partial paths. And you learn one model simultaneously over all these classes or different partial paths. And that gives you improved performance assignment, in particular for internal nodes of the tree we observed for some data sets. And it was also implemented a bit more efficiently, so it's faster.
So that means it tells you that something is at an internal node?
Yeah, it makes assignment to an internal node. We can also do this here, but here, basically the model for the internal node, the classifier for this assignment to this internal node, it shares parts. So the model for this class is partly the same than the model for this class. They share exactly proportionally. So the part of the solution that they share is proportional to the path that they share. So you get basically taxonomically consistent different classifiers, and that for some reason is better at internal nodes. I'm going to show you an example. Right, so we've been working on different collaborations, also with JGI. So that was Phil Hugenhaus was involved back then, and so we looked also at lots of data sets that had relation to bioenergy generation. So for instance, with Falk Warnecke as the first author of these termite data set, then the wastewater treatment plant, something from Yellowstone National Park, Lake Washington with allotrophs. Then we looked with Peter Turnbaugh at human gut samples of obese twins. And we've done several studies with Phil Pope and Mark Morrison, who are from Australia, initially on the guts of kangaroos, then from the guts on reindeers. And now we're looking also at bioreactors. And with the Max Planck Institute in Cologne at the moment, we're working on different kind of plant metagenome samples. So they're studying the plant microbiota. So this is this example that I just mentioned, where we compared the two different tools. What you see here is Phil Pope. So he sequenced a metagenome sample from the microbial community in the gut of this Australian kangaroo. And he gave us training data for wallaby group 1. So he gave us a few contacts he suspected to come from this novel organism that he termed wallaby group 1. And there were no related genomes available. But with that few little training data, you could build a composition-based model for Phyllopaecia and Phyllopaecia S, and then assign the contacts. And what you see here is every little box is a contact. The green boxes here are those assigned to wallaby group 1, and they're scaled according to their lengths. If they're in one row, they come from the same scaffold. And so, and the lighter colors are partial assignments on the path to wallaby group 1. So maybe just to Succini vibrionaceae or to a parent of that. And red is inconsistent, so that's wrong, like going down a different path. Now, assuming these things that are on one scaffold, they're supposed to come from the same genome. So much green in one row is a good sign, and having red in here is bad. And we call this measure, so how much was assigned basically to the correct taxon or to correct higher level taxon, the scaffold contact consistency. And both tools are good in terms of that, so 89 versus 97%. And, but what you see here now for Phyllopaecia S is that instead of the red ones, you have more lighter shades of green. So you get more of these partially consistent predictions. That's presumably due to the better way that it's modeled. And then, so what Phil did was he used this draft genome reconstruction, and he found genes in there for nitrogen degradation and some antibiotic resistance, and he thought that this would provided evidence that he could cultivate this isolate on media containing starch and what he called urea as a nitrogen source, and he also added this antibiotic. And then he got this wallaby group 1 in pure culture, and was able to sequence the draft genome, sequence the genome. And when you compare that with the wallaby group 1 from the metagenome that we recovered, there was around 99% sequence similarity, so it was definitely the same organism. And comparison in terms of completeness, this was 90% versus 85% of the draft genome data was found in the metagenome bins. So there was a good kind of validation that this thing picked up something, that phylopaesia is allowed to recover draft genomes. Now this I'm going to skip because Johannes is going to talk about it. That's basically the idea is, so this is Johannes, so Taxator-TK is a toolkit. The idea is that if you use sequence similarities, then a common approach is to pick the top scoring homologs and the taxa that they are associated with. And so if that's your query, these OCABS could be unresolved subtrees in the respective taxa. And then you just assign the parent taxon in the respective reference taxonomy, the so-called lowest common ancestor. That's your prediction of what the query sequence Q also belongs to. And so how you define this group of taxa in the neighborhood is of course critical to what you predict. And so Janus came up with some smart way of how to do that. Because at the moment in many tools it's arbitrary. You just decide you go for the top 10 or the top 3, and that changes what your lowest common ancestor will be. And it could be too low, like this thing, or it could be way too high, like further up the tree. So now getting to the more biological things, I'm going to talk about the study that we did with the Max Planck Institute in Cologne. And so the senior PI on this project is Paul Schulze-Leffert. And together with, so he has a postdoc, David Bulgarelli, who's interested in studying plant microbiota. And together with David and 2 students from my group, so with Ruben and Philip, we looked at barley microbiome samples. And in particular, the topic of interest was is there an enrichment of particular microbes from the soil around the plants and the rhizosphere or even inside of the roots? And can we see some traces of what's going on there in terms of evolution? And this is something that hasn't been studied so far, I think, in metagenomics. So we were just curious if we could apply some techniques we used in other publications or in other studies into these kind of data sets. So Paul also he looked at 3 different kind of accessions, which is like plant strains, I've learned. And he wanted to see if they also differ according to how far they are evolutionary several years or several thousands of years apart. And he wanted to see this reflects different levels of domestication. He wanted to see if that also impacted the microbiota. And so he had this experimental setup where they sequenced from 2 points in time. They took biological replicates. And from the 3 different accessions, they got samples from the rhizosphere and from inside of the roots of barley, always for 16S sequencing. So that turned up in over 42 samples. And then also 3 metagenome samples just from the rhizosphere, but from both replicates, so 6 samples, and that was 45 gigabase pairs of sequence. And so they focused on the 16S part and compared the roots, the microbial taxa in the soil to those in the root, and found an enrichment of certain OTUs from certain taxa in barley. So these are the blue points here, and they're scaled proportionally to their abundance on a log scale. So these blue ones here are quite abundant. And it's interesting because they earlier did a study on Arabidopsis roots, and the red dots are Arabidopsis. So there are some similarities in the taxa that are shared across these plants, even though they're not that closely related. Then we could apply Taxator-TK to the shotgun metagenome datasets, and that does a taxonomic binning, but from the binning you can also derive a profile. So you can make an estimate of how abundant different taxa are by counting the assignments of reads. And so the advantage of that would be that we don't have this PCR amplification bias if you look at shotgun datasets that we introduce in the amplicon phase. And there are no copy number biases in terms one of them is 16S, so there's organisms that have multiple copies of the 16S operons, and then that of course sort of adds to the estimate. And so what you see here is a comparison for the different taxa of the abundance inferred from the metagenome to the abundance inferred from 16S, and the overlap in green. It's again on a log scale, so you can see the rare ones in comparison to the more abundant ones. And we see, and additionally, what you see are things that are not picked up by 16S, like archaea, so those you only find from the shotgun. And also there were a few eukaryotes that were detected from the shotgun data. And mostly there was a good consistency, but phyla that are known to are under-amplified with 16S, such as cyanobacteria and chloroflexi, so we see more of them being picked up with Texator, so from the shotgun data. And phyla that have multiple copies of 16S, they are somewhat less abundant in the shotgun data. So that kind of made sense together. And if we compared it quantitatively, so we had 16S, the amplicon survey, and the numbers from that. And then we also identified 16S from the shotgun data and classified them both with the same method, and then correlated that and got a correlation coefficient of 0.89, so quite good. But the differences here are likely due to these primer amplification biases. And then we compare to the assignments to the metagenome bins, now this time using Texator. These differences would reflect both primer biases of the 16S and variations in the copy numbers of the RNA, because both of these things don't affect the metagenome bins. And again, the correlation, it was just slightly lower than this, so it seems primer bias presumably is more a stronger influence on changing the results than the RNA copy numbers. But it's nice to see that there is overall good coordination between these, or a correlation between these different samples. Then what we did next was we found the certain taxa that were enriched in the 16S study around the plant. So that seemed to be plant associated in the root. And we could map those or identify the respective bins in the shotgun data. And then we identified from the functional annotations of those bins whether certain functions are more abundant in the bins relative to the rest of the samples. And that returned in this list of functional categories. And what we found very interesting is that many terms in here related to interactions with the host. So if you have bacteria and hosts, they have these arms races, so the pathogen with a plant immune system, and the secretion type system type 3 is important for that. Also processes, I think, such as adhesion and these sugar phosphorate transferase systems. Then also we find things that relate to viruses, which is interesting, and to bacteria interaction. So this is where this system is assumed to be important for. And some things also that, also bacteria interaction. The siderophores are these common goods resource in studies of bacterial interactions. So it looks from that what you find as enriched around the plant are things that relate to arms races between the host and the plant, and maybe also between viruses and bacteria. And
well, you can't show that and not show the opposite of what is enriched in the soil and not in the plant.
we didn't look at that, what's enriched in the soil, honestly.
Yeah,
We're interested in what's enriched in the plant. I don't know, I can ask.
You might get exactly the same list.
Huh?
You can get the same list.
No, you can't get the same list, else it wouldn't be significant in one direction.
I think you should look.
Well, we could, sure. Okay, and so the next thing we did was look for positive selection in the metagenome samples. So positive selection, you can search for that by looking in protein coding sequences if they're enriched in non-synonymous versus synonymous mutations. So non-synonymous means it changes the amino acid and therefore the protein. Synonymous is silent, so it doesn't have any effect. So we assume it's more random. And if something is enriched in non-synonymous mutations, this usually happens if there's an advantage for it, right? So it's selected to change. And that's usually the case in arms race scenarios. For instance, if you have an antibody targeting a particular protein, it's beneficial for the respective pathogen to change, so it's not recognized anymore. And the similar system going on for plants interacting with the effector proteins of the particular bacteria. And so how we did this was we mapped reads on gene families and then removed repeat rich families because these would be possibly more ambiguously mapped, inferred trees from that data, and then on the trees mapped on the branches where non-synonymous and synonymous changes occurred. And you do that to account for correlation introduced by relatedness of different sequences. And then on these reconstructed numbers of synonymous and non-synonymous changes, we performed significance tests. And so this is then what we came up with, is that there were indeed a significant, or a fraction of those samples, of those metagenome shotgun samples, where had protein families with significant evidence for positive selection. And looking at the terms, we saw again in red those little stars here. These are just the top 30 of the list. We found many that related to phages, but also things that related again to pathogenesis and secretion, such as the type 3 secretion systems, and also to some so-called effector proteins that, so these are the proteins that are actually are assumed to be interacting with the plant. So few of these have actually been characterized in depth. So you rely on predictions of tools there also, and they have distinctive signals, I think, in the N-termini that are used for that. Okay, right, now I already used up my half hour, I think, so I'm going to end on that and not speak about this topic, but would like to thank the people in my lab that have worked on this. So I mentioned a few already. Also, what I didn't talk about are these two guys. So they're looking at predicting the plant biomass degradation phenotype, and also using statistical learning methods and trying to identify protein families that are relevant for making a bacterium or other kind of microbe a plant biomass degrader or not a plant biomass degrader. And these are our collaborators. So for this project, I showed these two were particularly very important. And yeah, thanks.
Have you ever looked to see if the membrane proteins, if you're getting positive selection on the inside or the outside?
Yeah, we looked at that. So we have some things where we looked at whether it's transmembrane inside and outside. We found it slightly enriched on the outside, as you would expect. And also what was interesting was that so these proteins that are targets of the plant immune system are usually highly conserved in bacteria, but in these highly conserved overall proteins, there are part regions that are under positive selection strongly. So these are good targets because the bacteria cannot get rid of them. But then there are some parts where the immune system then recognizes it, where they can change, and they seem to have usually shorter stretches of residues that are under positive selection. And so we looked for such stretches, and we could find that also. So under the proteins that were under selection, there were some that had these stretches of residues, clusters in the sequence, where they seem to have more mutations.
Maybe a related question, but were you able to distinguish between directional positive selection or diversifying selection? So, you know, in an arms race, you might expect that you could just diversify in any direction. But
you mean towards a certain direction, or
in any direction, just diversity is good.
Right? Yeah, it was just diversity. Yeah. No, we didn't distinguish between that. Yeah, just diversifying. Yeah.
You should get positive selection. Just diversifying doesn't help you, right? Because you still have to maintain function. Things that are under selection. Does that make? Am I right about that? Don't they have to run and maintain structure?
I mean, they can change in certain regions, and that's where you can see this enriched rate of non-synonymous substitutions. And so, we haven't looked whether it's particular changes that are accumulated preferentially, but just that it seems to be regions that are higher, more variable in terms of accumulating. And then others, of course, have to be they're constrained due to functional constraints, things like that.